Hello Bio 220 students, and welcome to Online Microbiology. For this first learning module, there are going to be 3 PowerPoints, 3 core slideshows of lesson material, but they're only in addition to the required readings from the textbook. Well, this first PowerPoint, which will be in the lessons portion of Module 1, a lot of it is just going to be definitions and remembering terms. So we won't go through all 106 slides in one video, or together I should say, but we will go over some parts of it. Well, first thing about microbiology is this is a study of microorganisms, which are just things that you're not going to see unless you have something to assist you, like maybe a microscope, something that magnifies the vision. And these include a variety of examples with a variety of functions. But common examples are bacteria, fungi, protozoa, which would be microscopic animals, as well as microscopic algae, and even going into viruses as well. And, well, something to note, not all microorganisms actually cause damage or are disease-causing, but those that are, they're called a very specific term, pathogenic. Now, a variety of microorganisms, they do things that are important, like help decompose waste or fallen leaves or dead plants, as well as provide oxygen. Most of the oxygen we breathe, actually, are provided by microbes. And then as well as help assist in providing vitamins, say because of microbes in our gut, or helping to prepare food, like preparing vinegar and cheese and bread even. Yeast helps with bread in that scenario, helps it rise. There's also a variety of pharmaceuticals and other things that are produced thanks to microorganisms, one of them being insulin. Now, some other things to note. Well, denim fading can be caused by trichoderma, and well, basically this is just to show that even in the manufacturing sector, microbes are very important. Reason why microbiology is important is because the more that we understand about microbes, one, the better we can help, say, prevent getting diseases from more dangerous ones. The ways we can better prevent diseases, the way we can better prepare food to prevent spoilage, and well, just the more we know about the microscopic world, the better we're able to help our own level of living and our products of care. Alright, naming and classifying microorganisms. Essentially, there's a way of organizing the vast variety of microorganisms in order to be able to logically pair up different groups and different sets of microorganisms into similar qualities. And well, this sort of scientific naming, scientific nomenclature, developed by Carolus Linnaeus. And 2 things to make note of. Every organism that is given a scientific name has a genus and a species. And basically the order that they show up is it will be a Latin term where the first word is capitalized, and that is always the genus of that species, and the second word, which is lowercase, is always the species. Now, essentially, they're just really helpful in helping to describe different organisms. Let's take E. coli, Escherichia E. coli, for example. Well, the Escherichia, that's given credit to the person who first discovered that organism, and then coli just indicates that it shows up in the colon or the large intestine. So these names oftentimes either describe where they're from, what function they do, or what general family of organisms, collective of organisms, these things are related to. Now, Staphylococcus aureus, just another example. The Staphylo means that it's a spherical type of organism, and the Coccus means it's composed of cells. The Aureus part, well, they're gold colored. So again, these scientific names, they do describe sort of the organism, giving an understanding of what the scientist or researcher or doctor, clinician is dealing with or looking at. And well, again, just for, say, any future assessments, just know that the first word of a scientific name is always the genus, and the second word, which is lowercase always, is going to be the species. Getting into types of microorganisms. Well, there is a large variety, from bacteria and archaea to fungi and protozoa, algae, viruses, and multicellular animal parasites even. Well, essentially, these range from either single-cell or multicell cellular organisms, and we're going to get into each one. Well, bacteria, they're going to be an example of prokaryotes, which means they do not have a nucleus or a brain of a cell, a individualized compartment called a nucleus. Now, bacteria, they're single cell, and very specific to bacteria is that they have peptidoglycan cell walls. Peptido means it has a peptide, a protein, and glycan meaning there is a carbohydrate, sugar-based cell wall. This is very unique to bacteria. And well, they divide by binary fission, which means they simply just split apart to reproduce, not sexually, but asexually. And well, they derive nutrition, derive sustenance from the surrounding material, and also in many cases through photosynthesis, just like plants. Getting into archaea, very, very different from bacteria. And the reason is because where bacteria all have a peptidoglycan, a protein carbohydrate cell wall, archaea do not. And actually, their cell walls have a wide variety of composition. Something that archaea share, though, is that they actually often live in extreme environments. For example, say low oxygen environments that have a lot of methane gas. That's where methanogens, or organisms in the archaea domain, that like being in high methane environments. There are also extreme halophiles, which means that, say, on areas where it's very salty or almost completely filled with salt, where most organisms would die from the lack of water, halophiles are archaea that actually enjoy and thrive in that high salt environment. And then there are also extreme thermophiles, which are archaea that are very accustomed to extreme temperatures that would actually kill most other organisms. And these can even be found in, say, underwater vents that are exposing either magma or highly heated gas near the bottom of our oceans. So, archaea, very important. Their cell walls have a wide range of composition and is not as concise or consistent as bacteria, and they thrive in extreme environments. Whereas archaea and bacteria were prokaryotes, so things that do not usually have their own organelles or nucleus. Now we're going to get into eukaryotes, which are organisms that do have a nucleus, that do have a brain in their cells, or I should say, who do have concise, discrete packets of organelles within them, sort of like how we have organs in our bodies. Eukaryotes have organelles that do specific functions in their cells. Fungi, well, their cell walls are unique in that they're made of a substance called chitin, and, well, they absorb organic materials for energy, usually by decomposing dead or decaying matter, like fallen trees, animals, roadkill, things like that. An example of fungi would be yeast, which is a unicellular or single-celled version of fungi organisms, or fungi microorganisms. And then another example would be molds and mushrooms, which can be multicellular, consisting of multiple cells working together. Now, another type of microorganism would be protozoa, and they're also eukaryotes. They have a nucleus in their cells, and they can do two things to take in organic chemicals: either absorb it across their membrane, just like many other organisms, or actually, in a way, ingest it by engulfing them in their own way. They may be motile via pseudopods, cilia, or flagella. Motile, M-O-T-I-L-E, essentially means that it's an object or an organism that can move on its own. If something was mobile, M-O-B-I-L-E, then it has the ability to be moved. Like, say, a box is mobile, but a box cannot move on its own. If it could, it would be motile. So that's the difference between mobile with a B in the middle and motile with a T in the middle. One can be moved, and the other one can move of its own accord. Now, protozoa, they're free-living and parasitic, and they derive their nutrients from some living host. What these slides have been showing is essentially the scale that many microorganisms are on, which are micrometers, or μm. Now, another microorganism are algae. They're also eukaryotic. They have a nucleus. Their cell walls are composed of cellulose, whereas fungi were made of chitin, and bacteria were made of peptidoglycans. They're found in all sorts of water sources, as well as in the soil we walk on. They use photosynthesis for energy, and they do actually help produce a good amount of oxygen that we breathe, as well as produce many carbohydrates, or sugars. They're on the large micrometer scale, going from 5 to 10, and now algae, which can easily be in the hundreds of micrometers. Viruses are a unique type of microorganism. They don't consist of cells, but some trait that is unique to all viruses is that they consist of two things. One of them is that they have genetic material, either DNA or RNA. And then the second quality that all viruses share is that they have a protein coat. Two other types of infectious agents would be viroids, which only consist of infectious genetic material, like DNA or RNA. And those typically are found in the plants, or typically affect plants. And then there are prions, which are infectious proteins, but lack genetic material. Those are things that usually only affect animals. But viruses, unlike viroids and prions, they consist of both genetic material and a protein coat, and these can be found in the animal kingdom and the plant kingdom, infecting a variety of hosts, even fungi. If it has a cell, there's probably a virus that infects it. Now, the outside protein coat, it may be enclosed and in a lipid or fatty envelope, and, well, the way that viruses replicate is by hijacking the internal machinery that a cell uses to replicate. And, well, they'll essentially invade a host cell, replicate inside that cell, and then go on to infect other cells. They're not on the micrometer scale, they're typically on the nanometer scale. Now, there are another type of microorganism for microbiology is concerned. These consist of multicellular animal parasites. So parasitic animals that consist of either a few or many cells. They are eukaryotic. They do have a nucleus. They might not all be microorganisms. but some that are talked about are flatworms and roundworms. And well, your reading material should go on even further into their life cycles. Now, essentially, these main microorganisms have been broken up into three main domains, which are bacteria, archaea, and eukarya, which eukarya actually consists of not just microorganisms, but also macroorganisms like ourselves. The reason for doing these three different domains is because most eukaryotes, they are very similar, even if they're only a single cell to multicells, and they're more closely similar to archaea. And archaea are much different from bacteria, even though both archaea and bacteria are prokaryotes. Now, next few slides, we're going to be getting to some big names. Robert Hooke, he is one of the people who proposed that we consist of essentially little units, or what would eventually be called cells. And this essentially is the process of beginning the process of cell theory, which is an important theory in biology that simply states that living cells have to come from already existing living cells. The first microbes that were observed by Anton van Leeuwenhoek were called animalcules, which he needed to see through a magnifying lens. So getting into some different initial beliefs into how cells or how living things originated. There was one initial hypothesis, which was spontaneous generation, and it was the notion that life just simply came from non-living matter, and that particular non-living matter had to just be something in particular that was needed for life. For example, decaying meat was needed to produce certain life, because any life could need meat or vegetables. Now, biogenesis, it was a hypothesis that living cells had to come from pre-existing living cells and did not just originate out of thin air. So, over many years, many different scientists proposed and performed different experiments to try to prove one theory or hypothesis over the other. One done by Francisco Redi was that he filled jars with decaying meat, and here's what he noticed. He noticed that jars covered with a very fine net did not have any maggots grow out of them. And he noticed that jars that were exposed to the atmosphere did have maggots grow in the meat that was contained. And then sealed jars that were perfectly sealed off from the atmosphere did not have maggots growing. So this is just starting to show that a prominent belief at the time was that if you had decaying meat, maggots originally came from that meat. But this is starting to show that maggots did not come out of thin air, and eventually people would learn that it was because flies were laying their eggs on decaying matter, and that it wasn't just because decaying meat was just a source of maggots, but it was a place where flies would lay their eggs. John Needham, he put boiled broth into covered flasks, and what he found out was that nutrient broth that was heated and placed in a covered flask did have microbial growth. Now, this wasn't necessary flasks that were perfectly covered and were probably still exposed to the air, which explains why microbes most likely were able to still get in. So that was an inconclusive experiment. Lazzaro Spallanzani, he redid that experiment, but he did it quite differently. He really boiled the broth, killing anything that was already in there. And then afterwards he sealed it and then heated it some more. And what was found was that there was no microbial growth. Therefore, this supported, or I should say, this disproved the theory of spontaneous generation, because if all that was needed for life to occur was the starting material, then even though the flask was sealed and heated, after it cooled down, microbes should have grown again. So, in some cases, scientific theories and hypotheses may not be proven directly, but the opposite can often be disproven. Now getting into the theory of biogenesis. Rudolf Virchow, he said that cells arrive from pre-existing cells. And, well, something that Louis Pasteur did, he demonstrated that microorganisms are present in the air. And what he did in his experiment was he had one condition where he had broth that had a lot of nutrients in it, and it was placed in a flask that was exposed to the environment. And it was found that microbes did in fact grow in that exposed flask of broth. But in a separate vial, he had a flask of broth, the same broth, that was heated and immediately sealed, and no microbes grew in. So again, this is showing that just because you had the nutrients needed for life, life did not necessarily grow out of those nutrients. So this is another thing that is disproving spontaneous generation. And then Pasteur also used S-shaped flasks, which was essentially a way to keep microbes out but let air in to the flask. So those flasks were not completely sealed off, but they were designed in such a way that microbes would have difficulty traveling up into the flask. And what this showed was those flasks that had broth in it and the S-shaped caps, no life grew in them even though they were exposed to the atmosphere. Essentially, the neck of the flask trapped those microbes, and microorganisms were shown to definitely be in the air, but they could be kept from going into, well, basically what this is showing is that there are microorganisms in the atmosphere, and they don't just originate out of nowhere. Okay, a golden age of microbiology. Well, this was essentially a few decades where microbiology really became a defined field in science. It began with Pasteur's work, and his discoveries included the relationship between microbes and disease. That disease was not necessarily some mystical force, but there were usually, or it could be proposed that there were microbes at work causing those diseases. He also found different ways to have immunity and make antimicrobial drugs to prevent infection. Pasteur showed that microbes are responsible for fermentation, or the process of converting sugar to alcohol when there is little air. Microbial growth is also responsible for why many foods and beverages go bad and spoil or become rotten. And it was also discovered that bacteria that use air will spoil wine by turning it to vinegar, which is essentially they're producing acetic acid. Pasteur demonstrated that these spoilage bacteria could be killed by heat, and that was not hot enough to evaporate the wine or alcohol. So essentially a way of getting rid of the disease-causing agents in drinks to make them safer for consumption and not causing microbial diseases. Pasteurization became the term for essentially applying heat to kill off harmful microorganisms in substances that we would consume. So essentially the main takeaway of Louis Pasteur's work, he demonstrated that life did not arise spontaneously out of nowhere or from non-living matter. Augusto Bassi, his work showed that silkworm disease was actually caused by a fungus, so fungi that is a microorganism. Pasteur showed that another silkworm disease was caused by a protozoa, so again showing that microorganisms are things that exist, even if we can't see them without equipment to advance our eyesight. And then Ignaz Semmelweis advocated hand washing to prevent the transmission of fever or puerperal fever from one obstetrical patient to another. Now, applying Pasteur's work showing that microbes are in the air and can spoil food and cause animal diseases, Joseph Lister used a chemical antiseptic, which was phenol, to prevent surgical wound infections, so helping to disinfect tools and, well, help people by getting rid of microorganisms that could invade the body during injury. Now, a milestone in the golden age of microbiology. Well, this is Joseph Lister's work, performing surgeries with aseptic conditions using phenol, basically disinfecting the region that was being worked on. And it proved that microbes caused surgical wound infections and were something to try to get rid of during medical operations. Robert Koch discovered that a bacterium causes anthrax and provided the experimental steps known as Koch's postulates to help demonstrate that a specific microbe caused a specific disease. So this was the start of the medical field gaining a foothold of understanding what causes diseases, after which it would become better to understand how to prevent diseases. Now, when it comes to vaccination, this is starting with the work of Edward Jenner. He inoculated a person, or essentially infected a person, with a cowpox virus. And after that initial infection, that person became immune to smallpox. Essentially, what that was showing is that under certain conditions, one infection can help prevent the disease of another infection. Now, it should be noted that viruses range heavily, and so getting a vaccine for one particular virus might not prevent the infection of another type of virus. They're all very different. It just was luck of the draw that cowpox and smallpox were so similar that getting infected with a less serious cowpox virus helped prevent death from a more fatal smallpox virus. And well, these were actually originally done on cows and eventually used to help prevent humans from getting sick from deadly smallpox. Now, the buildup of protection from a virus or infectious agent through vaccination or other ways is called immunity. In early times, there was a belief that there would be some magic bullet or some way of treating diseases to help prevent infection. And well, chemotherapy was developed to treat diseases with chemicals. Therapy with chemicals. And then chemotherapeutic agents used to treat infectious disease can be synthetic drugs, which are made in a lab, or they can be antibiotics. And antibiotics are really just chemicals produced naturally by bacteria or fungi that inhibit, they slow down, or kill off microbes. Well, what this slide is basically getting into the magic bullet that Paul Ehrlich speculated could possibly exist. It was essentially a substance that would destroy a disease-causing agent, a pathogen, but does not harm the host, which was the desired outcome. Whereas many of us probably know how chemotherapy can really hurt the person that it's meant to help. In many cases, either hurting their skin or hurting their hair cells. Well, what was discovered was actually sulfonamines, which are very effective against certain infectious microbes, but doesn't exactly kill us, thankfully. There is also another situation of the accidental discovery of antibiotics. Alexander Fleming, he discovered the first antibiotic by accident. And Fleming, what happened was he observed that Penicillium fungus had a really good ability to stave off or prevent the growth of other infectious diseases, and this was called penicillin. This material that penicillium produced was called penicillin. And it was tested clinically and was shown with much success at helping build up cures or helping to cure people many different ailments. Well, essentially, one of the last things we're getting to in this long slideshow is going to be different fields of microbiology: bacteriology, mycology, parasitology, immunology, and virology. Now, three very common ones are going to be bacteriology, which is the study of bacteria. Mycology involves working with fungi. And then parasitology, which involves the study of parasitic organisms as well as protozoa, microscopic animals. Now, immunology, that is, again, getting into the fact that this first slideshow is going to be a lot of definitions. Well, there's immunology, which is the study of immunity, such as vaccines and interferons, how they're used or how they can be used to prevent and cure viral diseases. A major advance in immunology was discovered in 1993 by Rebecca Lansfield, who classified streptococci based on their cell wall compositions. Rebecca Lansfield, well, she discovered differences in the chemical composition of the polysaccharide in cell walls of many pathogenic streptococci, basically showing how infectious agents can have a variety of carbohydrate-based cell walls. Virology, which is very well connected with microbiology, is the study of viruses and also other infectious agents such as prions, infectious proteins, and viroids, infectious genetic material. Dmitry Iwanowski in 1892, as well as Wendell Stanley in 1935, discovered the cause of mosaic disease of tobacco plants was actually the cause of a virus. And well, something very specific, electron microscopes have made it possible to study the structure of viruses in detail. Viruses are very small, much smaller than bacteria or cellular organisms. Therefore, while there are a variety of microscopes, electron microscopes have the greatest ability to magnify an image. We can see the smallest things that, of all the things that microscopes can be used for, the smallest things that microscopes can observe are from electron microscopes. And well, recombinant DNA technology. These involve microbial genetics, which is the study of how microbes inherit traits, molecular biology, which is the study of how DNA directs protein synthesis, or essentially how our bodies take our DNA in our nuclei and convert it into proteins that actually provide function, either for our genes or helping us maintain levels of temperature or how to digest different materials. Genomics is really the study of genes in a particular type of organism, and recombinant DNA is DNA made from different sources, like from one organism and another organism, or a plant and a bacteria, combined to help provide help in one particular case or another. Recombinant DNA technology involved a variety of, scientists who worked along the way, slowly building up from how genes encode for cell enzymes, all the way to how DNA is what is the hereditary or the inheritable part of our code, and how mRNA or messenger ribonucleic acid is sort of an intermediary between DNA and protein synthesis. And, a timeline of the golden age of microbiology. Microbial ecology. It's the study of the relationship between microorganisms and their environment. Sometimes bacteria converts oxygen or carbon, as well as a variety of other elements into forms that plants and animals can use. Whereas initially, perhaps how phosphorus naturally occurs, our bodies as humans may not actually be able to utilize. Bacteria in our gut can convert that into a usable form of phosphorus, rearranging it, putting it into a different compound, things like that we can now use. And bacteria can be used to degrade organic matter in sewage to help clean up impurities and pollutants in water. And that's what most municipal plants actually use to help clean waste and actually clean water supplies. Now, the remainder of this long slideshow is going to consist of many more definitions, and we're actually going to get into the other two slideshows very, very soon. But just continuing on. Let's see. Yeah, to be honest, much of this, it'll just be add definitions and terms. Normal microbiota. What normally belongs in your body can be called a flora or a collection of microbes. And this term has evolved into the new term microbiota. Microbes normally present in and on the human body, they're called your natural or normal microbiota. Let's see. Several types of bacteria. Your normal bacteria, which your body typically has, they help prevent the growth of disease-causing bacteria, mainly because if you have your healthy bacteria, they're the ones who are consuming a lot of the nutrients in your colon that you can't absorb, therefore preventing disease-causing bacteria from reproducing and growing. A lot of these normal bacteria in our bodies, they help produce vitamins in our body, like vitamin B and K, as well as there is a resistance, which is the ability of our body to ward off disease. Our normal bacteria, our healthy bacteria in our gut, they help ward off infectious bacteria. And we have many of our own resistance factors, which is like our skin or our stomach acid. to help either prevent disease-causing agents from getting into our body through our skin as a layer of protective covering, or whenever we eat something that has, say, infectious agents in it, our stomach acid typically does a very good job of really nuking whatever is in the food that we eat. But also the antimicrobial chemicals that we can ingest or produce. Something particular is biofilms. These are typically produced by bacteria, and they're essentially what you'll see on your teeth if you see a film that you can scrape off on your teeth. That's actually from bacteria grown on your teeth in your mouth. And well, essentially biofilms, it makes it harder to reach the actual infectious agents. So that's why it's good to actually remove those. Many EIDs, or emerging infectious diseases, they're simply new diseases that have come into recent light, or diseases of increasing incidence, reaching either large localized population or global infection. One example would be Middle East Respiratory Syndrome, or MERS, a very nasty situation, which is a type of coronavirus. It's a common version of, it's common to SARS, which is severe acute respiratory syndrome, and 100 deaths in the Middle East between 2012 and 2014 due to MERS. It belongs to the same family of viruses as COVID-19. There are infectious diseases called avian influenza A, which are primarily in waterfowl and poultry. Well, they can be transferred from human to human, or I should say, the potential of transferring them from human to human has not yet been observed, but it is possible that a virus may mutate someday to gain the ability to go from one person to the next. Now, the remaining slides, these are going to be various additional material for you to look at, essentially getting into the shape of different infectious diseases. And well, to be honest, this is getting onto slide 100 of 106 slides. So really the key here is that there are a lot of infectious causing agents that are microorganisms, and something to note is that they're all called pathogens. Well, that concludes the first lesson. We'll get into the next lesson video shortly.